remarkable study published in Science Advances says life on Earth has two origins instead of one. The study by evolutionary biologists at the Institute of Molecular Evolution at Heinrich Heine University in Düsseldorf, Germany, looks at a time in Earth's history, some 4 billion years ago, particularly at what is known as the last universal ancestor of all cells, or LUCA. Evolutionary biologist Dr. William F. Martin, senior author of the study, says, and I quote, "The new data leave only one conclusion. The bacterial and archaeal lineages made the transition to the free living state independently. Only free living cells are alive. Let's call it by name. We are looking at one origin of the genetic code, but two origins of life." Unquote. Professor Martin spoke to MCR from Düsseldorf. Welcome to My Entire Reports, Dr. Martin. It's a great pleasure to have you.
It's a pleasure to be here.
Many congratulations
Thank you very much.
to you and your colleagues on an extraordinary study. I want to start by trying to understand if there is a distinction between life having two origins and it having started twice, because I'm a bit conflicted by the headlines in some of the media outlets.
Well, when you work on early evolution and the media start discussing it, many things can can go haywire. It's not haywire though. So what we've got here is we have probed a phase of evolution where metabolism was still evolving. Okay, metabolism meaning enzymatic metabolism. All right, so all cells have to make amino acids, bases for RNA, and cofactors, vitamins, in order to grow, in order to perform the basic processes of life, making proteins, replicating. So what we have identified here are a set of about 420 reactions that convert gases on the early Earth into amino acids, bases, and cofactors. It's 420 chemical reactions. Many of those reactions take place in our cells today. They're largely universal.
I see.
But the enzymes, okay, so the reactions are one thing, but those reactions require enzymes. Enzymes are biological inventions. They're proteins. They're products of the world of protein synthesis. So what about this? What is the evidence for a single origin of information processing? There is incontrovertible evidence to support the view that the information processing system that we use today, that all cells use today, is of a single origin. It's the genetic code, right? The genetic code is very complex. All cells use the same code. All cells translate nucleic acids into proteins using the same kind of ribosome, and they all use the same. So there is one origin of protein synthesis. Now, what have we found? We found that if we probe, so we probe this phase of evolution where we already had protein synthesis, a ribosome, and it's starting to invent enzymes. Now, what enzymes will it invent? It can invent anything. There is no directed evolution or any agency in any of this, okay? It will invent what it can. But if that primitive genetic system, information processing system, invents enzymes that feed back into its own synthesis, that is, that help catalyze reaction to make its building blocks for the ribosome and for proteins, then that leads to what are called autocatalytic cycles. There's an autocatalytic feedback, and that helps the process to go forward. Now, the question is, those 420 reactions, were they all present in the last universal common ancestor? Yes. Were the enzymes that catalyze those reactions present in the last common ancestor? The answer is no. What we find is we find a process for lineage-specific divergence between the bacteria and the archaea from a universal common ancestor that had the genetic code that could invent proteins and was what was metabolically incomplete. It could not synthesize its own components. And that's why we have evidence for that independent process of enzyme invention leading to free living cells. So now we get into a definitions issue. Are we calling this last universal common ancestor that, as far as we can tell, was living in rocks? And we'll get into that in a moment. Do we call that, is it alive? And our answer is no, because it's not a free living cell. It is not self-sufficient. It requires inorganic catalysts that were present at the site where metabolism and life arose as precursors for the enzymatic reactions that later came along to replace those inorganic cofactors. So the last universal common ancestor was the last universal common ancestor was dependent upon its environment for fundamental biochemical processes, catalysis, that enabled it to exist. Free living cells, bacteria and archaea, once they emerge, they're independent. They can grow on gases: H2, CO2, ammonia, H2S, phosphate, and water, and they can grow. So that is genuinely free living. When they escape, then the life process is complete. The origins process is complete. We have free living cells. Before we get to that phase where there are free living cells, LUCA is not alive. It can't support its own existence. And therefore, we can speak of one origin of the genetic code, and we have evidence for two independent origins of cells. And because we define free living cells as alive, it's two origins of life. So that's okay.
Oh, you're okay.
I'm okay with that.
Okay, now, Luke, would you describe LUCA then as sort of an intermediate state to life?
Yes, it's an intermediate. There was a time on this early Earth, on the early Earth, where there were no cells, there was no life, and then there was a time where there was life. And in that transition, there were things that were half alive. LUCA is such a construct. It was half alive. It had the genetic code. It was synthesizing proteins.
Yeah.
Amazing. Okay, so imagine this chemical reaction contained within rocks that's synthesizing enzymes as an intermediate state in route to life. But it's not free living. It's dependent upon its environment. It's half alive. It can do a lot, but it can't. But it cannot conserve energy all by itself. It's dependent upon sources of carbon and energy and nutrients from the
so
from
that
the Earth.
Begs the question: the leap from that half alive, LUCA half alive, to bacteria and archaea. What is that process? It's astounding to me that leap happened, right?
Well, if we're honest, these are actually the first insights into this phase of evolution that anybody's ever obtained. There have been many studies that have focused on LUCA, but they have worked under the assumption that LUCA was a free living cell, and that either bacteria evolved from archaea or archaea evolved from bacteria. And you won't find very much literature on that process because it doesn't work. They are so different, fundamentally different in their basic biochemistry, as we're uncovering here, and in the way that they compartmentalize themselves from the environment, that it's extremely unlikely that bacteria evolved from archaea or vice versa. The simplest solution are two independent origins. Now, your question was, refresh my memory.
No, I was talking about the leap from half-alive LUCA to
bacteria and archaea.
Yeah. Okay, right. And so we're getting the first insights into this this process of evolution where LUCA is half alive into the alive versions of bacteria and archaea, identifying a new protein invention that happened in these two lineages. We're also seeing the finalization of the ribosome. Bacteria and archaea have different collections of ribosomal proteins on their ribosomes. It's about 30 that are conserved and about 20 that are added in the two lineages. And so we can see that addition. We can see a process from a primitive ancestor to a more advanced ancestor in the bacteria and the archaea, and those processes took place independently.
What kind of material did you study in order to reach the conclusion that you've reached? I'm curious, did you actually study physical material to arrive at this, or it was more like an intuitive understanding from the data available?
Okay, there are three kinds of data that underlie this study. One is genomic data, and information from genomes, the presence and absence of enzymes. There's also structural data, because sequence comparisons to see whether certain enzymes are widely distributed across all lineages in life. That the sequence comparisons have a certain level of sensitivity, but structural comparisons are much more important, because there are many sequences, there are many pairs of proteins where the sequences are not related by normal criteria, but the structures are almost the same. So this can happen. So what is more conserved in evolution are structures more so than sequences. So we incorporated structural data to get better insights into this phase of evolution. And we also incorporated chemical data, chemical reactions. And this is something. If one of our figures plots the reactions of metabolism, 420 reactions of metabolism, and about 40% of those reactions have been shown in the laboratory, either the exact reaction or a very similar reaction, to be catalyzed by metals that exist in hydrothermal vents. So it's three kinds of information: genomes, structure, and chemical reactions.
No, it is fascinating to me that you basically, in a sense, looking at Earth 4 billion years ago, right?
Yes, that's so the question is, how do we know that life is 4 billion years old, roughly 4 billion years old? There are several formations on the early Earth preserved in rocks that are 3.5 billion years old. Those are what are called they're fossilized microbial mats, indicating the growth of microbial systems growing. They are from, interestingly, hydrothermal systems. They're not photosynthetic. They're hydrothermal systems. Strelly Pool is one such example, if somebody wants a quote on that. And there's also evidence for biological methane being produced on the Earth 3.8 billion years ago. So somewhere between the origin of water on the Earth, which was 4.2 billion years ago, and the origin of methane, which is evidence for methanogenesis or archaea, we have an origin of life. That's a window of about 400 million years in which the first cells evolved. All things considered, it's a very short period of time.
I'm glad you're saying that, because it almost seems as if early Earth was in a desperate rush to create life. I mean, to me, it sounds like that.
Well, that's one read. Okay, I'm a scientist, so we can't. As a scientist, we can't say there was an absolute need to hurry up and create life. Otherwise, this, that, and the other is going to happen. There are schools of thought that will say life is unavoidable.
Right.
It's inevitable. Okay, there's that school of thought. I fundamentally disagree. Death is inevitable. Life is an opportunity. Okay, so there's We know that there's when the sun engulfs the earth, and the life process will have come to an end here. But life is an opportunity. So what this does indicate, though, is that if it could happen that quickly on Earth, it could happen that quickly elsewhere. Mars is a typical example. While it was still wet, its conditions were very similar to the early Earth. And the same kinds of reactions could have been going on. Whether we can still find evidence for such reactions is still up in the air. There's a moon of Enceladus, a moon of Saturn called Enceladus, where hydrogen is being produced and water is being spewed out into space. Those are reaction conditions where the kinds of chemical reactions that primitive organisms use to gain energy and to make their cell material are taking place. So if there are the argument then is if reactions on the early Earth that generate hydrogen and native metals, and that these hydrogen and native metals plus CO2 make the basic building blocks of life. If you incorporate ammonia into the equation, then if this works on in the laboratory, which it does, many people have not noticed this. We can put hydrogen and CO2 ammonia in test tubes with native metals. That's iron and nickel and cobalt, and we can get amino acids and very interesting compounds generated from simple chemical energy present on the early Earth. There's no sun needed for all of that. So what we're identifying is a kind of environment where it's possible, but it is not a requirement. And actually, one of the things that emerges from this is that it might have been easier to get to a LUCA state than it was to get to the free living state, because at the free living state, you have to have at least 1,000 genes packaged into a thing of the size of a cell, and it needs this cell needs to be able to harness energy from the environment in a way that it becomes self-sustaining, becoming independent of the energy of the early Earth. That gets us into phosphate and phosphite, which is different.
Yeah, the next
question.
This may be a bit ignorant, but I'm going to ask you nevertheless. There are two lineages that we are talking about: bacterial and archaeal. Now, life as we know it,
Yes.
you and I, for instance, what lineage are we? Oh, we are both.
We're both.
Oh.
We're both, because we rose from symbiosis. We have mitochondria. The mitochondria in every cell of our body are the small organelles that generate ATP, the energy currency of cells. We humans generate about a body weight of ATP every day, and that's because our mitochondria are consuming oxygen to burn amino acids and fats and sugars that we take up with our nourishment. So a well-nourished human makes about a body weight of ATP every day, and that is the job of mitochondria. Mitochondria are once free-living bacteria, and they entered into an endosymbiosis, that is one cell living within the other, with an archaeal host about 1.5 to 1.8 billion years ago. And that was the origin of the eukaryotic lineage, the lineages that have nuclei, complex cells like our own. It was also later in the plant lineage, cyanobacterium came in to become the chloroplasts of higher plants. That's why they're green and why they can photosynthesize. So we sit on both sides of the early branches of the tree of life.
This is somewhat of a tangent, but visualize for me Earth 4 billion years ago as an evolutionary biologist. What does it look like to you visually 4 billion years ago?
Well, okay, 4 billion years ago is after the moon forming impact. So at the very beginning, the standard narrative of Earth origins goes that the Earth formed by accretion, and that is just a collection by gravity around a solar disk of basically a protoplanet. In the case of the Earth, there was a collision. There was a fateful encounter with a Mars-sized planet called Theia, a theoretical object, but it's pretty well documented. that impacted the Earth, that melted the entire Earth, converted all the carbon into CO2, which is excellent because that's what all ecosystems start from today. They start from CO2. It melted the Earth. It gave rise to the moon. That impact also gave the Earth its 23-degree tilt, and converted all the carbon on Earth into CO2. The rain, the oceans rained out or were also extruded from the Earth as it cooled. That gave rise to oceans that were probably, according to many estimates, about 10 kilometers deep. And that's why that's twice as deep as today. Why is that? It's because there's about an ocean volume of water retained in the magma and the crust, and that comes from these interactions between water circulating through the crust, generating these hydrothermal vents. And it's not just a little bit of water, it's a lot of water, okay, about an ocean volume. So the oceans were very deep. By many accounts, there was no land on the 4 billion year old Earth. Okay, they're the oldest sedimentary rocks that we know, meaning evidence for erosion. They're 3.8 billion years old. So it was a pretty barren place, and there was nothing that was really alive. The sun was much fainter, and the surface was probably not. So the surface of the ocean there was not a lot going on. But at the bottom of the ocean, in hydrothermal vent settings, there could have been quite a lot going on. One of the interesting implications of this theory is that if these first cells that emerged in our theory, they're hydrogen dependent CO2 reducing organisms. Today we call them methanogens in the archaea and acetogens in the bacteria. These cells emerged, they could populate the Earth and occupy niches. Well, what niche would they occupy? They would occupy the crust. They'd go back, be drawn down through circulation back into the crust where they get exactly what they need: metals, hydrogen, and CO2, and they populate the crust. That process could actually go very quickly if you think about it. I can give a quick calculation. If you take a bacterium today, a laboratory bacterium like E. coli, Escherichia coli, and you give it unlimited growth substrate, and a good temperature, 37 degrees, it will double every 20 minutes, and that culture will outweigh the Earth in 48 hours.
Yes.
Okay? Yes, that's bacterial growth. The trick to that calculation is there's no trick to it, it's real, is that it's 144 doublings. We'll call it 150 doublings, okay? So that's 2 to the 150th amounts of new, that's a very large number. And so if you have a generation time of 20 minutes, it takes 48 hours. If you have a generation time of a year, it takes 150,000 years to occupy the Earth. Okay, so the process, once life takes off, there's no turning back, and it can occupy the niches very quickly. That leads to another question. Very often get this question, if I may say so. Dr. Martin, could it be that there were other forms of life that also arose? Or could it be that if we go down into hydrothermal vents today, we can see this process going on? So the answer to both questions is probably not. And the reason is that the niches were occupied very quickly by living cells. And so anything that reached the level of a LUCA was a rich substrate, but had not escaped. So that's going to be devoured by these carbon-starved organisms that are populating the Earth. And the same thing is today. At hydrothermal vents, what we can see is evidence for formate synthesis, conversion, reduction of CO2 into formate and reduction of CO2 into methane. And those are the same processes that sit at the base of microbial metabolism. So we can see evidence for the same process going on, but it doesn't go all the way to the origin of new cells because there are already cells there and they're eating all the good stuff that process might synthesize.
That is a remarkable statement which say that I'm quoting now, because this is your statement. metabolism is a
Yes,
non-negotiable property of life. It transforms. It is.
it's a good sentence.
It transforms.
Yeah,
Exactly. It
it's a
jumped
good
out at me.
sentence.
I'm a connoisseur of great sentences, so yeah, it immediately caught me. Now, it described to me. I mean, I understand broadly what metabolism is, but for my viewers, since it's so fundamental to life, describe it a bit for us.
So metabolism is this, life is on the bottom line, a chemical reaction.
Right.
Okay, we eat food, we breathe air that releases energy in our mitochondria that gives us energy to go, and then it also gives rise to, through chemical reactions, building blocks, right, for muscles and for skin and for hair and for protein, the basic substance of cells. So those core reactions that produce these basic compounds, amino acids that all the proteins are made of, bases that all the nucleic acids are made of, are information processing, and the cofactors, the vitamins that we require in order to make these reactions work. All organisms
Right.
require vitamins. Some of them we can make ourselves, some of them we get from our nourishment. So those basic reactions, how complicated is it to make those compounds, those 20 amino acids, 18 cofactors, and 8 bases of RNA and DNA from gases, from H2CO2, H2S, water, ammonia, and phosphate? How difficult is it? Well, it's 420 reactions. It's a very finite set. In our metabolism, it's even less because we don't make half of our amino acids. We get them through our nourishment. So we have a smaller set of those metabolic enzymes. And many organisms also get their amino acids, their protein, or their cofactors from their diets. But these organisms that make everything from the gases, they're a living collection of these ancient enzymes and genes, because they use these reactions to grow. And so we can study that. We go out and look, what are they doing? Okay, and the answer is they're using 420 reactions. And then we can start, and then we can say, okay, well, how old are the genes for those 420 reactions? And it turns out that about half of them go to LUCA, the last universal common ancestor that was half alive. And the other fourth here and fourth there are invented on the way to bacteria and archaea, underscoring the deep divergence between the bacterial and archaeal lineages.
You are.
Am I making sense?
Absolutely.
Am I making sense?
Just last couple of things, Doctor. You have mentioned that LUCA possessed enzymes for only about half of the reactions of metabolism. The other half was catalyzed by metals in the environment where LUCA arose. Now, what if those metals were not available in that environment? What would have happened?
Nothing.
Yeah.
Okay, no, this is what happened. I'll give you an example.
Okay.
I'll give you an example. We can take here's a little sample reaction tube that we run in the laboratory on a day-to-day basis. Okay, it's a 5 milliliter vial, and it's got about 1.5 milliliters of liquid in it. And at the bottom, there's this black stuff, and that's a metal catalyst. Okay, now. All right, so those are the kind of reactions we do in the laboratory. So if we put hydrogen gas and CO2 gas into this vial with water and no catalyst, nothing will happen for a day, for a week, for a year, for 1,000 years. Nothing will happen. It will not react at all. If we put the right catalyst in there, nickel or iron, or palladium or cobalt, these are metals that are found in hydrothermal vents today in their native state, right? We're talking about, shiny metals like this spoon, okay? The spoon is the shiny, but we're talking about shiny metals. That's what those are the good catalysts. If we put a shiny metal in there, then the hydrogen and CO2 will react on the metal to form formic acid to make acetic acid, or pyruvic acid, which is one of the most central compounds in the metabolism. Is still the pyruvate is what our mitochondria import today to synthesize to gain our energy during our metabolism. So the catalysts are essential. Without the right catalyst, nothing happens. So my answer maybe was short, but it was correct.
Sharp and succinct. Do you believe that life is intrinsically rare because of the number of variables at play, or do you think life has a way of emerging easily anywhere in the universe?
So you're asking me, and this is a clear opinion question. And with beliefs, okay, the scientist has belief. We have things that we believe to be true for the sake of argument. Those are called premises that we have to structure our scientific research. So how likely, you're asking basically, how likely is it that life arises? Well, let's see. We got to LUCA. We have an observation set of one on this planet. There was one universal common ancestor. So it's possible on a sample of one, it's possible as one. And that the idea that last universal common ancestor could only give rise to two free living descendants, is actually naive. Probably it was possible within the hydrothermal vent environment that we posit for where we see these reactions happening, that many different lineages could have emerged, but only two made it. Okay, so it could be that the process of packaging 1,000 genes into a small package that has everything, absolutely everything that you need. You can't forget anything, okay? Everything that you need. And that packaging process gives rise to a cell that can also replace all the energy input that was being delivered by the hydrothermal vent, can replace that all by itself just by using enzymes. That might be a limiting principle. So the two origins of life might seem like a lot, but actually, if we think about what was possible, we might be looking at a limiting principle en route to a populated planet.
Finally, to conclude, this is a sort of a broader theme, which is perhaps outside the realm of what we are talking about, but I'm going to ask nevertheless. You have addressed it a little bit early on. Was the emergence of life incidental or inevitable, given the chemistry of the early Earth?
Well, the chemistry of the certain reactions are probably inevitable. Okay. Certain reactions, chemical reactions, hydrogen reacting with CO2, that happens in lots of different laboratories independently. That's highly reproducible. That kind of reaction is going to take place. There's no question about that. But whether it advances all the way to life or not, like I said, life is not inevitable. Death is inevitable. Life is an opportunity. So it's possible.
On that note, Dr. Martin, I want to really thank you for your time. It was fascinating for me. I hope it was worth your while, because I was coming from a lay background, although I have studied physics and chemistry, but nothing at the level that you operate at. So I hope it was okay for you.
I hope it was okay for you. It's biology, and so as a final statement, let me just say, if we look around at a university, I've been at a university for 30 years now, and you look around at your faculty, what you'll notice is that mathematicians typically do their best work by about age 25. Physicists typically do their best work by about age 35. Chemists did their best work 115 years ago, and biologists do their best work by about age 65, not because we're stupid, but because biology is really hard. It's not 92 elements with 8 oxidation states or a half a dozen laws that operate everywhere in the universe. it's a million species of beetles and each has its own exomicrobiome and
Yes.
endomicrobiome. It's complicated. But thanks to genomics, we're making progress in
All right.
understanding the
Okay.
process.